Alrighty, this video is for Unit 2, Question 1, or Essay Question 1. So for the start of this, we're starting with our alpha motor neuron. This is skeletal muscle. Our alpha motor neuron will have our buong, or energy, that comes down, and that opens our voltage-gated calcium channel. We also have some sodium channels over here. And we have these vesicles that are filled with acetylcholine here in blue. They have little blue polka dots all over in them. And those vesicles will carry them through the neuron, and through exocytosis it will release them here, where they will activate these N1 or nicotinic 1 channels. And this is the sarcolemma, so it will come in here. And that also has our buong that comes through here, our wave. We have our voltage-gated sodium and potassium channels here along this. It'll come down to here where our SR is, which is our sarcoplasmic reticulum. And the sarcoplasmic reticulum has a high concentration of calcium. And our energy comes down. This green square here that I have drawn here is our DHP. And then this little red spot right here is our ryanodine. And then here, this little blue square with the C is our calsequestrin. I hope I spelled that right. But what happens is that will come through, and then our calcium will be released very quickly. And this is our T-tubule right here. And yeah, the calcium is released. We also have a Circa pump on our SR, and that pumps calcium against its concentration gradient, so back into the SR, and that's powered by ATP. Let's see here. Next, we have our sarcomere. So this is the sarcomere. We're going to zoom into it. We're going to go over here and it's going to get bigger. But we have our nebulin, which are these straight lines going across here. That's our nebulin. These little squiggly lines is our titin. And then our myosin is here on our titin. And then, I don't know if you can see this very well, but we have our little red dots here. Those little individual red dots is our G-actin. And then that whole section, the whole chain of that is called our F-actin. So that's our chain. Here's what myosin kind of looks like if you zoom it in. We have the tail of the myosin right here, and then these are the heads of our myosin. So then over here, we've zoomed into our G-actin. We have all of our little circles, our red circles are our G-actin. And then these little areas right here are these little binding spots, which is where we have our troponin, which is this. It's the ITC. It looks like Mickey Mouse. The I binds to the G-actin binding site, which is these spots right here. And then our T spot binds to the troponin, or to the tropomyosin. And then the C spot has a high affinity for calcium. So calcium will come over here and bind to that. And when it does, it goes through a conformational change. It basically moves the tropomyosin kind of off our G-actin, off those G-actin spots. And that allows our myosin heads to connect here so that our muscles can contract. Here we have a table that kind of just summarizes our 1, our 2A, and our 2B. And it tells us where it gets its energy, how much it fatigues, how much mitochondria it has, blood supply, all that stuff. But usually the main source of energy for all of our muscles and our cells is ATP. We also have cytosolic ATP and creatine phosphate. Those are kind of our main sources of energy. Here's our crossbridge cycle. This is in skeletal muscle. This is the myosin head right here. And then, so we have our myosin head right here. It has a phosphate attached to it. We get calcium to come in. The calcium causes it to bind. And then we have a power stroke, and the power stroke causes us to lose the ADP that we have on the myosin head. Also causes us to lose the phosphate. It also releases some energy. And then this is our cocked state right here. And then we get ATP, which goes here, and then through hydrolysis we get that phosphate back. And then over here with smooth muscle, it kind of looks the same, but basically we have MLCK, we have an extra phosphate involved. ADP we still have that involved in kind of the same areas. And then right here, one thing that's different is that we have ATP. Oh, this is tonic, by the way. And we have ATP. ATP kind of right here where it says latch in this little section here. Our ATP will try to bind, but it won't be able to, and we'll try over and over until finally it does right here. It will be able to bind, and then again we go through hydrolysis just like we did with skeletal muscle, and we get that phosphate back. Tonic is like muscles that are sustained, so sphincters, those are contracted for long periods of time. This is kind of the cycle it will go through as tonic, so we definitely like having our sphincters working. Over here we have phasic. Phasic is like your eyes, or fast twitch muscles. It's like short duration or rhythmic. Kind of looks the same, but we have MLCK, yeah, kind of the same, because we have a phosphate over there that we get. We have MLCK. Our calcium, one thing to make note of is that it only comes from the ICF, or the SR, the sarcoplasmic reticulum. And we have our phosphate, this is ADP right here. I needed to use a different color, but that's ADP. The ADP gets released. We also have heat release, and we lose our phosphate also because of the power stroke, which is the same with the skeletal that we talked about earlier. We get our ATP back, or not back, but we have ATP come in. That will come here to the myosin head. We get our phosphate back. We have MLCP involved in this one. We lose that phosphate and it comes back to the starting point. You could also take this path where we have hydrolysis. We have our 2 phosphates here, and then we come back to this spot here. It's just a good way to help us see the differences between the two. And then the last thing we have to talk about is our Golgi tendon organs, our stretch reflex, and I think we have a few other things, but I didn't put them up on the board. But basically, let me pull up my notes so I make sure I say this right. We have, and muscle spindles. I didn't put that on the board either. Muscle spindles are proprioceptors that monitor muscle length. They're usually near tendon insertion. So kind of right here, you could say, this is supposed to be your patella, and this is your femur and your tibia. But this could be like where a bunch of tendons are, so you'll have muscle spindles there. They respond to stress, stretch, and they cause contraction. So they pretty much will keep your muscles from tearing your bones apart, which won't happen usually, thank goodness. But that keeps you safe in that sense. Your Golgi tendon organs, so the one that we do have drawn up here, they respond to tension. They're a safety mechanism, and they cause relaxation. There's where the stretch reflex is drawn. We have the bicep and the tricep drawn over here. But we have all of these different neurons and tendons and things. I think I'm going to add a few things onto here.